I'm coming from the clinical world, and I'm seeing patients every day, and genetic testing is very advanced in Israel. We'll get to that in a minute. So we have all kinds of tests done in pregnancy, prior to pregnancy, in children, in adults. And in the last, a year or two, I noticed that there are many dilemmas coming up from genetic testing done on either pregnancies or children with the assistance of gamete donation. And this is why I want to explore with you today. So gamete donation gives lots of hope to many families, but yet it poses many challenges, as we will see. And this is why I thought it's important to discuss this issue. But just to mention the expectations, I'm not going to give many answers, but I'm going to raise dilemmas and questions by using some example cases that I'll show you. I will be really happy to hear your opinions about these cases and questions. I will show you some preliminary qualitative results from a study that we're doing in Israel. And I'm happy to have collaboration if you find this interesting. So when we're talking about donor conception, there are three main ways: either a sperm donation, an egg donation, or an embryo donation. The main two are sperm and egg donation. And in Israel, the main one is sperm donation. So we do use egg donation as well, but we mostly import the eggs from outside of Israel. So it makes everything much more complicated. So I will mainly focus on sperm donation, but I think the questions are very, very similar. So in the UK, the Human Fertilization and Embryology Authority oversees the regulation of gamete donation. And this is a figure that you can find on their website, saying that since 1991, where they started to collect the data, more than 70,000 children were born with the assistance of gamete donation. So we're talking about big numbers, and this is the UK only, it's about the US or other parts of the world. This estimation is from 2008. I believe it's even bigger now, but every year 400 new sperm donors are needed to match the expectations or the needs of sperm donation. So again, we're talking about very big numbers. Becoming a donor is a very long process. They have to move a few steps. They have to have all sorts of tests until they are approved to serve as donors. And part of the process is genetic testing. And as you will see, it's becoming more and more complex over the years. The completion rate, any idea how many are reach the final destination of becoming donors? Do you have an estimation in percentages? How many of those who start the process will actually become active donors? Okay. So it's between 3 and 20, depending on the country, but it is quite low, right? So many people will start, but only few people will complete this long process. Another thing that is important to remember is that becoming a donor is only the beginning. Okay, it's a very complex process. It takes time, but then there are many years to come where people will use sperm. Okay, so once someone is becoming a sperm donor, our samples can be used years after the actual donation, years after the sperm donor doesn't give sperm anymore. Okay, and in every single moment, we can find babies born from this sperm donor, we can find children, we can find adults, we can find pregnant women, okay? Which makes it a very unique setting, okay? Very different to having a family with someone, okay? And that poses even more challenges, as we'll see. The current guidelines in the UK were issued in 2019, and what they say is that what made them change their guidelines after 2008 is that due to the rapid evolution of genetic technologies, the wide availability and uptake by the general public for genetic ancestry, key recommendations are made on information provision around this to all parties involved in the nation. So they do mention genetics, but it's mainly the genetic ancestry that made this change, because now people can go back and identify their sperm donor. Okay? And this is what made them change the guidelines. Whereas what I think should be is this. Here recommendations are made on genetic screening of potential donors, because we have more and more tests that we can do now before donors are accepted to be donors, and the long-term access to donors' DNA. I'll show you examples in a minute, and recontacting past donors for new genetic testing. So I think these are the challenges today, not only identifying sperm donors, which has changed in the last few years. So if we go back to the initial point of someone becoming a sperm donor and that genetic process that precedes this starting point, it does says on the guidelines that a donor should have, so donors should be asked about their family history and about their medical history to make sure that they do not have any serious genetic predisposition or genetic condition, and they specify which conditions exactly should be ruled out before someone can become an active sperm donor. Various diseases, epilepsy, asthma, hypertension, psychosis, as you can see here, and also a significant dominant Mendelian disorder, okay? And not any dominant disorder, but it should be according to the list of the HFEA for PGT. So anything that is approved for pre-implantation genetic testing is considered severe enough, and our sperm donors or potential sperm donors should be rejected if they have any of these conditions. And it's a very long list that you can find here. And also a chromosomal rearrangement that may result in unbalanced gametes. This is why all potential sperm donors are having a test called karyotype to make sure that their chromosomes are balanced and you don't have any unbalanced abnormality that may increase the risk of an unbalanced in their offspring. In terms of screening for genetic diseases, any recessive genetic disorders, and in egg donors also X-linked disorders, the UK guidelines only mention these 5 diseases for men, and this and another one for egg donations. So only very few conditions. These are the most common hereditary conditions around the world, according to the WHO. And this is why these conditions have been chosen. But is this enough? Is it okay that we only test donors for 5 conditions out of the many possible genetic diseases that we have? Looking at just, a week ago only, which is all the currently known disease-causing genes, we're talking about more than 26,000 disease-causing genes, okay, that we know of today. And of course, the more we test for, the more diseases we test for, the more carriers that we find. Okay? And I have some numbers for you. They're based on quite small studies, so it might be slightly different, but it shows the trend. So if we screen for 10 genes, and this is all done on potential sperm donors. Okay? So if we screen for 10 genes, we will find nearly 9% carriers. If we screen for 47 genes, which is the what they do at Cryo International, and this is the largest sperm bank today in the world, it will result in nearly 18% carriers among potential donors. And if we screen for 200 genes, they reach a number of 56%, which is quite too much, I think. But you can see that the more we test, the more we find, okay? The more carriers that we find. And then what will we do with all these potential donors that are found to carry recessive disorders? Okay, so they themselves are healthy, but only if the recipient is the carrier as well. then they have a 25% chance of having an affected child. Okay, you're talking about autosomal recessive inheritance. So the common practice in many places is to reject these potential donors. Okay, they're perfectly healthy just because they're carriers. This is what is done in countries, again, including the UK. What I think and what people have suggested might be better is to test the donor and the recipient and only reject or at least inform cases where both are carriers with the exact same disease, because we're all carriers of something. The chances that two people will carry a faulty and exactly the same faulty gene are much, much smaller. Right? But then it's more expensive, it's harder to do, but this is one possible solution. Okay, so we're starting at karyotype testing of all potential sperm donors, some sort of carrier screening, whether it's for 5 diseases or 47 diseases. We're talking about very concrete tests that are done for potential sperm donors as of today. What will become more and more available in the future is that donors or potential donors will have much more advanced genetic testing. And it can be chromosomal microarray analysis, looking for deletions or duplications of in our genes, or whole exome sequencing, or even whole genome sequencing. Okay? So this is definitely the future. It's not going to take long before we get there. And in Israel, it's already the present. Okay? And this is why I think we should think about these issues and find solutions. So in Israel, the largest sperm bank is part of Cryobank, International Cryobank, but they have their own branch in Israel, and they have donors from all over the world. And they offer two options. Of course, it costs more if you choose these options, but it's available to women. So they have the premium donors who have an expanded carrier screening test of about 300 genes. And they have the gold donors who have whole exome sequencing done, again, prior to them becoming donors. And of course, when we move for targeted testing, even if it's for 300 genes, to whole exome sequencing that covers thousands of genes, there are all sorts of results that we can come across. These are the 3 main challenging ones, for me at least, or from my experience. So there are conditions with incomplete penetrance, meaning that not everyone who carries a faulty gene will necessarily develop the disease associated with this disease. Sometimes we have percentage that we can get, 10%, 20%, 30%, and sometimes we don't even have a concrete number to be. Just know that not everyone will be affected. We have the variable expression and variance, meaning that some people may have certain symptoms, whereas others may have totally different symptoms. I'll give you an example soon. And then we have the group of the late onset diseases. Okay, so a BRCA1 gene defect, or any of the dominant diseases that can only express when someone reaches his 20s, 30s, and even more. So what do we do with all these findings if they are identified on someone's exome sequencing? What do we do with this potential donor? And another complexity is what type of variants will we disclose to APS to donors themselves? Is it only the pathogenic variants, meaning variants that we're absolutely certain that will cause a disease? Or shall we also disclose VUSs, meaning variants with uncertain clinical significance? So where do we draw the line of what type of findings we disclosed or not? Okay, so this is another interesting question. And it's important to bear in mind that we have a few people involved in this decision-making process. So we have the donor themselves, okay? This information might be relevant for their own health, for their children once they choose to start a family of their own. Of course, it's relevant for recipient people who want to accept the donation, and for children conceived with the assistance of this donation. And sometimes the interest of these parties conflict. So sometimes we don't not all these three groups have the same interest with regards to what findings are disclosed. And we as clinicians are somewhere in the middle, and we need to decide what to do with all these complex variants that we come across. So Julia Kaplan is the genetic counselor who's now doing her PhD, and she is collecting, she's interviewing clinicians, and she will develop a questionnaire later on. And she is trying to understand what are clinician attitudes towards the complex questions around donation or genetic testing in sperm donors, and what factors affecting their use. I will show you some results based on only 8 interviews so far. So of course, it's only preliminary results, but you can see the trend. And she has already interviewed 4 sperm bank directors, 2 genetic health professionals, a genetic counselor and a medical geneticist, and 2 fertility experts. And we're intending to collaborate with Professor Maya Sabatero from Columbia in the US and collect data from the US as well, and then compare between Israel and the US. So let's start with this case, okay? So everything here, again, are things that we came across in clinic. So I met with this man who came after he contacted a bank in the US for an egg donation. And he had a family history of her sister affected by a very severe genetic condition that was identified via whole exome sequencing. It's de novo, so it's new in her. It's not inherited from any of their parents. And he was absolutely adamant that he wants the egg donor to have whole exome sequencing, knowing from firsthand what does it mean to be affected by a genetic disorder. So lots of correspondence between me and the bank in the US, and the donor has agreed to perform whole exome sequencing, okay? And in this whole exome sequencing, it was done in our lab, by the way. They sent blood over to Israel, and we're the one who did the test. A genetic change in a gene called TANCA1 was identified. What do we know about this gene? We know that it causes intellectual disability, and also it's a risk factor for schizophrenia. The donor herself is perfectly healthy. At least this is what we know. She doesn't report a family history of psychiatric disorders or intellectual disability. And what's also what makes things even more complex is that when we try to classify the variant, this is what we got. So it's a variant with uncertain clinical significance, but it's very close to being likely pathogenic, okay? At least based on bioinformatics tools that we now use. Okay? So it's highly suspicious, but we can't tell for sure that this is a disease-causing variant, okay? A, because she is healthy, and 2, because it's not yet classified as pathogenic. We don't have experience from other people in the world having this change, knowing that they're either affected or not affected. So remember I spoke about variable penetrance, a variable expression, either intellectual disability or psychiatric disorder. And incomplete penetrance, it might be that someone is healthy, but we don't know for sure that the offspring will be healthy as well because of incomplete penetrance. What do we do? Okay, so we're at the lab, and we have this information now. Do we disclose it to the bank in the US? Will we disclose it to the potential father? Will we disclose it to the donor? Okay, what shall we do? Are you happy to raise your hand for your opinions? Or not so much? Yeah, okay, so whoever wants. Do you think the variant should be reported? Anyone? Yes?
Consented.
Is that good?
Have you been consented?
Yeah, absolutely. Yeah. There was a very, very long discussion about it prior to testing. She wanted to have all possible information that's available. And yeah.
But wouldn't have been specifically this.
Yeah, absolutely. Absolutely. Just in general terms, of course. Yeah. Anyone thinks no? Not sure? So, just to say that we did disclose it eventually. Okay. It was a long discussion also with the genetic counselor back in the US. We did disclose it, and the father did choose to continue with this egg donor. But should the donor be rejected now that we know about this genetic variant in her? Okay, should she be rejected from future use of her egg? Okay, I will not ask for your opinions, but this is another important question that the bank needs to give answers. And so a bit, some results from our interviews with clinicians in Israel, what we do see is that clinicians want maximal genetic information on donors in advance before they become active donors. And I have a few quotes. I want to know everything in advance. I want no surprises during pregnancy. That's a sperm bank director. There are plenty of donors. There is no reason why a sick child is born if we could have prevented it. Another sperm bank director. We as doctors want maximal information, simply put. I would choose EXO, the maximum test, so we can know that everything that is in our power to test has been tested. This is a genetic health professional. Eventually, all donors will do EXO. That might be the truth. And another genetic health professional says everything, CMA, exome, and when genome is available, these two. So maximal information whatsoever. I liked this quote coming from a sperm bank director saying that when CMA is now, at least in Israel, in the last 10 years, this is the standard test done in women having amniocentesis during pregnancy, and it looks for chromosomal aberrations, so either deletions or duplications. And he said, when CMA has become the standard test in pregnancy, I decided to perform CMA on all donors. My colleagues warned me that we will lose donors, but I think that learning about abnormal findings when the woman is 18 weeks pregnant is worse. So the more we know in advance, the less trouble we have during pregnancy and after the child. This is the major opinion of clinicians in Israel, at least the ones that were interviewed so far. They did mention two pitfalls of using advanced genomic tests such as exome, one being those dominant conditions that I gave you an example of, something that the donor hasn't known about prior to donation. So not that we know about a condition in advance and we reject a donor based on a medical information that is already known, is we've been hearing that we learn something new via genetic testing and that it has implications for the donor's health and future potential offspring. So this is one pitfall. And the other one is the issue of variants of uncertain clinical significance. I will not normally inform women of USs, only if this is the advice that they'll receive from the geneticist, just as the case that I'm showing to you. When we ask clinicians, again, we did interview donors, but when we asked clinicians, their impression was that donors themselves want to know as well. There's no gap between clinicians' attitudes and potential donors' attitudes. So as sperm bank director says, we added a sentence to the consent form saying, I'm interested or not interested to receive medical information that is relevant to my or my children's health. They all wanted to know. All potential sperm donors in his sperm bank. Only one donor chose not to know, but phoned me a few days later saying that he had thought about it and changed up his mind. Okay, so their experience is that people do want to know. Another sperm bank director says they even like having broad information. This way, when they want to start their own families, they may have information that they may not otherwise have. So they get very expensive tests for free that they can use when they start to have their own stem. So we spoke about the current moment of finding a genetic variant in a potential donor, but what about the long-term use of this donor, okay? Or what about past users of this donor that she had already if there are already children conceived with her egg. The guidelines don't give us much information about that at all. The only thing that I found is about donors' responsibility that they should inform the sperm bank if they develop any medical conditions in the years to come, any new diagnosis in either themselves or a family member. And with regard to the sperm bank responsibility, they say in the guidelines, clinic staff are expected to monitor the use and clinical outcome of donation. They need to know if any affected children were born. This is particularly important in sperm donation when if donors are not successful in generating live birth, they should be discontinued. Equally, if any adverse outcomes are reported from treatment, the use of the donor should be paused until this have been adequately investigated. Okay, so if the sperm bank learns that there is a potential disease, then they should stop using this sperm. So this is the only thing that I found in the guidelines about long-term implications of tests done after someone becoming a donor. Let's look at another example. So this is another family where a child was born with the assistance of sperm donation. And at the age of 11 years, so 11 years after the donation, he was diagnosed with developmental delay. All children, in the UK as well and in Israel, all children who have developmental delay undergo a test called CMA, so the test for deletions and duplications of the DNA. And a large deletion has been identified in the child that could explain his phenotype. We're not sure that this is the explanation, but this is a possible explanation. The mother has been tested negative for the deletion. Okay? We always like to compare the genetic makeup of the children to that of the parents, because if a genetic change is inherited from a parent, we will think that in most cases it may not explain the phenotype. There are exceptions, but in general, this is why it's really important to compare it to the parental DNA. And we also know that 10 other children were born using the donor's parents, and they're apparently healthy. So no one has reported developmental delay or any other medical issue in 10 other children. And again, testing the donor will allow better classification of the variant, is it pathogenic or not, and will also be important for other offspring, right? Future offspring that may be conceived with this sperm. And another thing is that we had a frozen sperm sample from a few years ago, for more than 10 years. You asked about consent, and we have the consent form of all donors who donated in Hadassah, in the hospital where I work. And on the consent form, it's a very old consent form, it hasn't been changed for years. On the consent form, it says, I hereby agree to perform any future test in my sperm aimed at determining whether a fetus or a born child is at risk of an anomaly or genetic disease, including genetic tests, according to medical advice given to the recipient. And they need to mark, I would like to either receive or not receive the test result. Okay? So looking back at this sperm donor and his consent form from 14 years ago when he became an active sperm donor, he marked the receive option. Okay, so he wanted to be informed of any genetic changes that are found in him. Just remember that the genetic world 14 years ago looked completely different from the way it looks today, right? Even 5 years ago, I would say. So what shall we do? What's the best course of action now? Okay. Again, we have a frozen sample of the sperm, and we can extract DNA from this sperm sample and test it. So, shall we perform CMA on DNA extracted from the frozen sperm sample and inform the donor only if positive for this deletion found in the offspring? Or shall we contact the donor and invite him for counseling and test him only if he agrees to be tested? Shall we simply assume it's de novo, it's new in the child and has no implication and leave it as is? Or we don't know. So we had to make a choice because we had this child that we wanted to find his diagnosis. But theoretically, anyone thinks that we should just test him and let him know only if he is positive for the deletion? Okay. Contact the donor and invite him to counseling. Assume it's de novo and leave it? Or not sure? Very thought. Already to prepare for that.
The donor and only testing as a control. So you were assuming that if he tests positive, not positive result for him, because he's a donor and he hasn't developed presumably hasn't got any of the phenotype that's there.
So it's the first option, just not informed. This is what you mean. Yeah, okay. So just to say that our legal department thought that the first option is the way forward for us because we couldn't really tell what to do. We actually thought about the second option as clinicians. We did want to contact him and explain the situation, but they told us just test the frozen sperm and inform him if anything is identified. What if on the consent form, he would have chosen not to have a genetic information? Will that change our opinion? And so I will not ask you to vote again, but I think it's an interesting question of whether or not his choice 14 years ago makes a difference in terms of our way of practice today. Okay.
For intuition, but it's the question of whether it's a result for him. So he's consenting to get results for him or not. But here you're not offering him a result. You're offering him a result on his child.
Yeah.
Question.
But it might have implications for his children if he chooses to start a family, right? So he is healthy himself, but it might have implications for that. So in a way, it is for him as well and not only for the child. So just a reference for a very interesting paper for one of the two people who are sitting here and a few others asking this question exactly. Is it acceptable to contact an anonymous, in this case it's an egg donor, to facilitate diagnostic genetic testing for the donor of conceived child? So it's a very, very similar scenario where a child is born with the assistance of egg donation and developed a condition suggesting of an hereditary condition. And this child is potentially eligible to be tested via the 100,000 Genome Project, for which both parents need to be tested in parallel to the child. And what's different in this case is that the fertility clinic denied to contact the egg donor on the basis that she had indicated on her consent form that she did not wish to be contacted for future research. And she also indicated that she did not wish to be notified if it was found that she had a previously unsuspected genetic disease or she was found to be a carrier of a harmful inherited condition. Okay, so the clinic rejected the application or the request to test the donor based on her consent form a few years prior to the actual situation where her testing was really important for the child. So in this case, we can clearly say that the rights of the donor conceived child and those of the egg donor and potentially the parents are in conflict. But she didn't want to know. We now think the testing care will be helpful to the child and of course to his parents who are anxious to have a diagnosis. And what Annika and Rachel and others write in the paper, and I do encourage you all to read it, but I will only bring the bottom line, is that it would be ethically acceptable for the fertility clinic to contact the egg donor in order to explore the possibility of her being tested. So not they're still going to ask her, they're not going to coerce her to be tested, but not to rely on a consent form that was signed many years back with other technologies in mind and other setting or mindset of this donor, and to explore her opinions now, make a decision based on her current opinion. Instinctively, the decision to contact the egg donor may feel uncomfortable. I think we can all agree to that. We argue that this discomfort arises because we continue to rely on a narrow model of consent, the consent form that she signed, that is unsuited to technologies such as genomic medicine and egg donation. So we have so many more complex situations these days that we can't really rely on a choice done a few years back when someone was at a certain age, certain familial setting, and with a different technology in needs. So the issue of broad consent, whether or not donors need to decide in advance what type of findings they wish or do not wish to receive on themselves, and the option of recontacting donors for further genetic testing is a very big question now that we have all these new technologies. And again, some findings from our interviews. So a genetic health professional said, broad consent is easiest for us, especially as I showed you that everyone just want to know. Okay, so we don't have refusals normally. It allows us to do what we like without getting back to the donor again and again, meaning every future pregnancy or child born, because it can happen again and again. But people may think differently over time. Therefore, the right thing to do will be to contact the donor for each new test. So the easiest will be just to accept the initial choice that they made when they become donors. But the right thing to do will be to ask them again and again if this is an information that they're willing to have. Yet, this is not practical. You don't know if you'll be able to break ship. If you do, at what stage in life. So it's a very complex thing to do, even if it's the right thing. Another sperm bank director said, I think we need to inform donors of new tests. When if something is identified, we shouldn't necessarily ask, rather inform. So we don't need to discuss it in too much detail, but just tell them this and going to do that. And only if they refuse this, of course, we cannot force them. I'm pretty sure the donors don't have a clue when they sign the consent form about potential future consequences. They have no idea that they may receive information about themselves. This is a fertility. So again, the consent form is a very narrow thing that we don't necessarily need to rely on. Another example, again, a child wanted the assistance of sperm donation, presented with global developmental delay, hypotonia, dysmorphism, so things indicated a possible genetic explanation. Again, CMA was carried out, and they found a very small deletion associated with a genetic condition called pregnancies syndrome. It's generally a normal condition, we know about that, and indeed the mother and the donor were tested, and they both have a normal CMA. So here we do have the full picture. We know it's a new genetic change in a particular child. We know that the donor doesn't have it in his DNA extracted from a blood sample, okay? But does this information impact any future pregnancies from the donor? Now that we know that it is something new in a particular child. So as geneticists, we know of thing called germline authenticism, meaning that sometimes some of the gametes, either the eggs or the sperms, will have a genetic change that is not found in the blood sample. Someone can be perfectly healthy, have a normal test from his or her blood sample, but their gametes may have this abnormality. Then there is a recurrence risk of about 1% for most things. Some things, the risk might be even higher. But for the common things, we do think that the recurrence risk is about 1%. I don't know what's the practice in the UK, but in Israel, if you find a new change, the novel change in one pregnancy or one child, this woman will be eligible for amniocentesis in each of her future pregnancies to test the fetus and make sure that indeed he or she inherited the normal copy of the gene. So this is part of our practice in Israel, meaning that potentially it could happen again in other sperms from a particular sperm donor. Chances are very, very, very small, but it is possible. And had it been a couple, we would recommend them to have amniocentesis in each of their pregnancies. But it's not a couple, it's a sperm donor. So what do we do with that? So we notify all past recipients of these sperm donors, and the same thing. The first option is they should not be notified based on the very small chance of recurrence. The second option is that past recipients should be notified and choose what do they want to do or not choose. Or I will not ask you to raise your hands, but again, it's another question that we face. What about ongoing pregnancy? Sometimes we know that women are pregnant at the time where we discover that sperm donor is a carrier for a particular genetic variant. Should we notify all these pregnant women and advise to have testing during pregnancy or not? Okay, so it's another question, but it's slightly different from contacting people who are not presently pregnant. What do clinicians thought about this? Should we inform of pathogenic variants identified in the donor or an offspring? So as turned out by directors say, I'm in favor of transparency. I know it puts women into a lot of stress, but it's much worse not to disclose and find out later that a sick child was born. This is a catastrophe. A fertility expert says we must disclose all the information we have. It is our duty. Even if it's incomplete penetrance, we cannot hide information. It's a woman's right to know, from the director. And I think this really explains what can women do with this information. Women should know so that they could choose what to do. They can look for another donor, they can use pre-implantation genetic testing, or they can do amniocentesis during pregnancy. Okay? If they know, they can have the option. And this is a nice quote as well from my director, differentiating between women who already received sperm from this donor and women who haven't yet received sperm from this donor. When women already have a child from the donor, they're often inclined to keep the donor even if something has been found. So they want to have two children from the same father, really. We may recommend to change a donor, but they will more often choose not. Who am I to determine that a woman changes the genetic makeup of her child if she already has a child? It's for her to decide. Okay? So again, they're in favor of disclosing this information to women. Another really interesting question that I don't have an actual example, but I'm sure we will face these questions the more we use whole exome sequencing or more advanced genetic tests, is regarding the late onset condition. So we mainly think of genetic testing in donors or in pregnancy that is related to childhood onset diseases that we can identify in a potential donor or during pregnancy. But when we use broad genetic tests, we can sometimes identify changes in late onset genes, again, such as the BRCA genes or any other genes associated with risks for cancer or cardiovascular diseases, things that only start later on in life, not in childhood. So should we test for these things when we do exome on donors, and should we disclose such findings to donors, to recipients? So just to quote, sperm bank director said, I don't think that my role as a sperm bank is to ensure a woman that her child will be healthy at 40, not even 30. Fertility treatments are successful if a healthy child is born. So according to him, this information should not be tested, it should not be disclosed, and donors should not be rejected if they carry these genes that they haven't known about prior to becoming donors. Now, a genetic health professional said something slightly different. With all due respect to prevention, meaning that if we know in advance that a carrier has a gene that we may offer more screening or prevention, you would still want to save this from his child. Had it been your partner, you're already together. But with a donor, you may always do some of that. So what she said here, that again, there is something to do with this information. People can be screened, there is prevention. But if you can choose to have someone else, why not? Right? I think that's a very strong sentence, but that was her opinion. Participants really wanted to have guidelines to assist them in making all of these difficult decisions. It would have been helpful to have criteria for rejection of potential donors and a committee to discuss complex situations. This is from a generic health professional. Perhaps similar to termination of pregnancy, we need to have criteria based on penetrance, age of onset, etc. So potentially we can find, what are limits when we think about what to test, what to disclose, when to reject, when to contact past recipients, and so on. So to conclude, conceiving with the assistance of gamma donation highlights the complexities of genomic testing. So we encounter these complexities already when we test fetuses, when we test children, but I think it's even more complex when it comes to using donors. There's the whole issue of consent, what type of consent, and how do we relate the consent given in the long term? How do we deal with all these complex findings, incomplete penetrance, variable expression, uncertain significance? And what are all of these long-term implications of, again, recontacting people years after they have donated, or contacting women who used a particular donor? When do we do that? And how do we do that? There are multiple parties involved. Okay, there are the donors, the recipients, the children, us as clinicians, society, norms. Guidelines will be helpful. Empirical data will be helpful. This is what we're trying to get some. And theoretical exploration of issues at stake will be helpful, similar to what you did in your paper. I think it would be really interesting to look at these particular questions and develop them and try to help clinicians when they face this in the ad. Thank you very much. This is my email address. I'm very happy to hear from you. If you have any questions, any thoughts, any collaborations in mind. And if you have any questions now, I'm happy to answer.